All right, we're getting started. We are recording. I want to give you guys a piece of advice on tests after this first one. I would not recommend waiting until the night before or a day or two before your test to start studying. Well, I would review a little bit regularly. I would review my notes unless you are an awesome auditory learner. The only person that I've ever had take my anatomy class and never study and make B's was Zach Lish.
No.
Of course. And that's because he remembers everything he hears. Okay, so cells are composed of protoplasm, right? What's in your protoplasm? This is also easy.
Protoplasm.
Oh, what's in your protoplasm? Protoplasm is that it's that aqueous solution that has carbs, proteins, lipids, nucleic acids, inorganic salt. It's what's inside the cell membrane. Right? That's what your protoplasm is. Everything inside the cell membrane. I might remember that. That protoplasm is the liquidy stuff inside the cell membrane. So every cell has a membrane. Why does it have a membrane? Why does it need a membrane?
For letting things enter and not enter.
So it holds things in that need to stay in, lets things out that need to get out. The cell membrane, it separates it from the external environment and from other cells, right? The cell membrane, it transports molecules so that we can do what? What happens when we transport molecules in?
We produce
Protein synthesis takes place in there so that we can keep replenishing our cells, right? So the cell membrane is necessary to hold everything in place and so that we can get nutrients in and out of the cell, nutrients in and waste out of the cell so that the cell stays alive. Nucleus, most important part of the cell, right? Why?
it stores your DNA.
Stores your DNA. Why do you need DNA?
It makes you new.
But what else does it do?
Tells your
Make
cells
RNA. But what do you need? What does your body have to do all the time to stay alive?
Reproduce.
Replicate. Right? We have to replicate. So DNA helps. DNA and RNA are essential for replication. And so since it has all of your DNA in there, the DNA is on proteins that are arranged in a loose state, and when they shorten, they make chromatin. Chromatin is what does chromatin become eventually? Chromosomes? Chromosomes. Chromatin eventually becomes chromosomes. And that's where your DNA is. So your nuclear membrane, if you have a cell membrane, why is the nuclear membrane important?
To keep things out of the
Keep things from damaging your DNA, right? Keep it floating around in there healthy, help with replication. So it's a double layer structure. It allows materials to pass from the nucleus to the cytoplasm, and the outer area is continuous with the endoplasmic reticulum, right? So the inner, it has two layers. The inner layer protects the nucleus and lets it have nutrients. The outer layer makes sure that things stay out like they're supposed to, and also it connects to the endoplasmic reticulum. I'm trying to not hurry, but hurry just a little bit. When we go to the parts of the cell, it's a little longer, but I want you guys to have a little bit of time to look over your notes and ask questions if you have them for tomorrow. So your nucleus and ribosomes. The nuclei are small round bodies that contain ribosomes called RNA. That make RNA. Ribosomes aid in protein synthesis. They may be in independent groups or in large groups called polyribosomes. So what kind of RNA is there? There are three kinds of RNA.
tRNA,
Which is what?
transverse RNA,
Transfer RNA,
and messenger RNA.
messenger RNA. Sorry. What's the other one? Ribosomal RNA?
I thought it said it with an R.
Yeah, ribosomal RNA. So what does messenger RNA do?
It messages.
Well, what is it doing? Is it picking up the ribosomes?
Yes.
Messenger picks them up and carries them to the transfer RNA, which takes it to
to the nucleus, into the nucleus.
takes it into the nucleus where it becomes ribosomal RNA to help with protein synthesis, right? So you have three types: messenger, transfer, and ribosomal. Messenger RNA picks up the information or picks up the ribosomes. Transfer carries it where it needs to go to help it become ribosomal RNA, which is what helps with protein synthesis. What happens if we don't synthesize proteins in our body? We can't break them down. We can't heal and repair, right? There is a disorder, and I took care of a young lady who had this particular disorder, where her lungs were unable to break down proteins, the lung cells themselves. And so about every 3 months, she would have proteins and lipids and fats because it couldn't do any kind of the cellular respiration and the cellular digestion didn't happen correctly in her lungs. They would fill up with proteins and lipids. And she would basically be drowning in her own body fluid. So they would have to go in and do what's called the bronchoscope, where they stick this tube down into both lungs. They would suck all that stuff out, and then they would lavage them, which means they would wash them out really good. She had to have that done every 3 months since she was 2 years old. She was 18 when I met her. She would have to continue to go to Children's all of her life because that is a childhood genetic disorder, and it's only treated at Children's hospitals. Since she was 2, she had not been home for Christmas or Thanksgiving. She had been in the hospital every Christmas and Thanksgiving since she was 2. Not only that, she was 18 and she was about 4'7" or 4'8" and weighed 75 pounds. So it had impacted her growth as well. Because if you can't breathe and you don't have good gas exchange, it affects your entire body, right? Okay, so that's just one of those instances whenever RNA goes awry, what can happen to your body. All right, now this was going to take a little longer. We're going to talk about all of the different parts of the cells. The cytoplasm is the semi-fluid material that is important. It's where all the life function in the cell happens. And then you have all those organelles, right? We're going to talk about each one of those organelles. So who knows what the centrioles do? So they're 2 cylinder-like organelles found near the nucleus, right? What do they do? You guys remember what the centrioles do? What they're important for? Remember the little spindles? Y'all watched the little video of the cell replicating where the spindle fibers start separating out? Those would be your centrioles. Or those little fibers that start separating out for cellular replication.
Where do you write all this down?
So if we're talking about, you're going to need to know what the cell parts do.
So centriole is a fiber that is separated.
It's 2 organelles found there, they're near the nucleus, and that is the part when cellular reproduction happens. Those are those 2 little spindle fibers that separate in the near the nucleus to help make 2 new cells. Prior to them separating in that reproduction area, they're inside the centrosome. The centrosome houses the centrioles. No. So the centrosomes house those centrioles. Then you have the endoplasmic reticulum, and that is a really fine network of crisscrossing tubules in the that are connected to the nuclear membrane. So the endoplasmic reticulum connects to the nuclear membrane, and it looks like a crisscross set of tubes. There are 2 types. There's rough endoplasmic reticulum. Why is it rough?
It has ribosomes.
It has ribosomes on the outside, right? On the outer edges. And why, and what is important about ribosomal endoplasmic reticulum? It has ribosomes, what does it do?
Helps.
Protein synthesis, right? Ribosomes and RNA are always going to refer back to that protein synthesis for breaking down proteins and utilizing them. So rough RNA helps with protein synthesis. So what do you think smooth RNA does? It has a smooth surface. What do you think we produce in the cell there? Cholesterol. We produce cholesterol there. That's where it's synthesized to use to make triglycerides for use in the body or for us to help reinforce the membrane. Everybody knows this one. What's mitochondria?
Powerhouse of the cell.
Why is it the powerhouse of the cell? What happens there? What do we make?
Glucose.
We make sugar. It takes glucose and it breaks it down to what?
ATP.
Which is what?
Glucose.
Adenosine triphosphate.
Oh.
So ATP is what we actually use for all of our energy source, right? That sounds like us from somewhere else. But so ATP is we take glucose and the mitochondria breaks it down into ATP
Is it through glycosis?
through glycolysis.
You know what
Yeah, through glycolysis. And we will learn about glycolysis and all of that, glycogen production and all of that kind of stuff when we do the endocrine system. The Golgi apparatus, what does it do? Do we really know what it does? We just kind of guess, don't we? Yeah, scientists think that it helps synthesize, helps break down carbohydrates in the cell. Where do we have them the most abundant? If it breaks down carbs, where do you think we have them the most abundant? So where do we start? So that means it deals with digestion. We're going to have more of those in digestion maybe, right? So in our mouth, because we start carbohydrate breakdown in the mouth. So you're going to have more Golgi apparatus in the cells in the mouth, and the cells in the stomach, and the cells in the pancreas, because that's where our insulin and that's where glucose and insulin and glycogen is broken down and functions, right? Lysosomes. What are important about lysosomes? What does lys mean? Break down and kill, right? So they get rid of the things we don't need in our cell. If a cell starts to break down, it will go digest parts of it, try to repair it. If it can, it'll break the whole cell down. Now, lysosomes are kind of like this little sac. They also have another name. They're called a suicide sac. Why would they be called a suicide sac? They sacrifice themselves. Or if they were to rupture, it kills the whole cell and surrounding cells. So if one lysosome inside the cell, because it's kind of like the vacuum cleaner of the cell, cleans up all the dirty stuff, breaks it down, gets rid of it, holds it in that vacuum cleaner bag. But if the bag busts, then it kills the whole cell and sometimes surrounding cells, depending on what that cell does. Peroxisomes. Did y'all learn about peroxisomes? So I had to relearn about peroxisomes last year because I think I might have learned about them in life science because I had this teacher that was crazy in 7th grade. But I don't think I learned about them after that. I don't remember about learning about them in college anatomy. But maybe I did and just don't remember. So that's the membranous sac that contains oxidized enzymes. It's important for detoxification. So kind of like how you pour this is how I remembered it. I can remember being told to remember it in junior high. Peroxisomes sound like peroxide, and when you pour peroxide in a wound, it detoxifies a wound. It bubbles up. So that's how I remember what peroxisomes do. They digest fats, and then they destroy harmful substances. So you pour peroxide in an open wound to clean it out, right? What is the cytoskeleton? That's easy. That's an internal skeleton, the internal structure of the cell, right? It gives it support. The cytoskeleton gives it structure and support. hemostatic vesicles. They form when the membrane folds inward. Why would our membrane need to fold inward on the cell? And kind of make a pinching, and that's how I remember it. Pino is kind of like pinching. It makes kind of a pinching envelope kind of looking part of the membrane. Why would we need that?
Replication.
We need it some for replication. We also need it when big molecules try to come into the cell. So say we, say I have a cell that doesn't need a polysaccharide, a long sugar. Well, if that pinocyte works appropriately, that polysaccharide, even though it normally could go through the cell membrane, it stops it because it's folded in where it can't go through all the little channels that are on the outside of the membrane. What are cilia?
Little things that
Little hair-like projections on the outside of cells to help grab particles, right? Where do we find cilia in the body? Most important place we find cilia in our body. Respiratory tract.
our nose.
Your respiratory tract. It's most important in the respiratory tract. Why? What is it doing in the respiratory tract?
Keeps out like other bacteria.
It's catching dust and bacteria and all that kind of good stuff. Where do we find flagella?
Little tail thingy on everything.
So where in our body? Where is flagella? Is it normally produced in our body, flagella? Flagella is generally only on sperm. If you have something in your body with flagella, like in your intestine, you're in trouble. Flagella from a bacteria. There are bacteria that have flagella that will go in and start causing some really bad gastric issues. Usually coming from nasty water. Which is why when you go to certain foreign countries, they tell you don't drink the water. Giardia is one of the worst ones. I've seen children with Giardia and they are so sick. Okay, I do not want to start cell division until next week because that's a lot and we don't have tons of time and I don't want to stop that. So we are actually going to stop here for today. I'm going to let you guys have just some time to study. Once again, you have a test tomorrow. And then Friday, all you guys have to do on Friday is just to label a cell.